Good morning, everyone. My name is David Shurtleff. I'm the Deputy Director for the National Center for Complementary and Alternative Medicine. And it's a pleasure to welcome you to our fifth 2014 Integrative Medicine Lecture Series. And it's my great pleasure to introduce our speaker for today's lecture, Dr. Bin He. He is a distinguished McKnight University Professor of Biomedical Engineering, Medtronic Bakken Endowed Chair for Engineering and Medicine, and the Director of the Institute for Engineering and Medicine and the Center for Neuroengineering at the University of Minnesota. Dr. He's research interests cover a broad spectrum in biomedical engineering, mainly in neuroengineering, biomedical imaging, multimodal neuroimaging, and brain-computer interface. So quite an extensive list of research activities for Dr. He. I might note here that, importantly for NCAM, his team's brain-computer interface work demonstrates that mind-body awareness training, such as yoga and meditation, can help improve performance on brain-computer interface tasks, which is very interesting for us. Dr. He has published over 200 peer-reviewed journal articles and is the sole editor of the textbook entitled Neural Engineering. Dr. He is also editor-in-chief of the IEEE Transactions on Biomedical Engineering. He has received numerous awards, including the Outstanding Research Award from the International Federation of Clinical Neurophysiology and the Established Investigator Award from the American Heart Association, just to name a couple. Dr. He is a fellow of the International Academy of Medical and Biological Engineering, IEEE, the American Institute of Medical and Biological Engineering, and the Institute of Physics. Dr. He has served as past president of IEEE, Engineering and Medicine and Biological Society, the Society of Functional Source Imaging, and the International Society for Bioelectromagnetism. Most recently, Dr. He joined NCAM's National Advisory Council as an ad hoc member, and he also serves on the NIH Brain Initiative Multi-Council Working Group to advise NIH on research funding and future initiatives for brain. Today in his presentation, Dr. He will explore recent research on quantifying imaging pain using electroencephalogram and functional MRI, and mapping thoughts from electrophysiological and hemodynamic measurements. So please welcome Dr. He.
Good morning. Thank you very much, David, for the kind introduction and for the invitation. It's my great pleasure and honor to be here and to share with you about the research my student and colleagues in University of Minnesota and I have been doing in the past several years. So my topic is about how to mapping the dynamics of the brain. So this slide illustrates, first of all, our brain activity is not just distributed over the three spatial and dimensions, so we need high spatial resolution imaging, but also it is really a dynamic process, which is very important because we really want to map the information processing process, not just saying where the neuron is located. This slide just illustrates what out there, and as what we know that ideally we would be able to, we want to get this high spatial, high temporal resolution and recording, but these single unit or multi-unit activity local field potential are all invasive approach. The blue line shows the various non-invasive imaging technology as dominant neuroimaging modality as a functional MRI, which is very precise in spatial domain. However, it is limited in its slow response in the time domain. If you look at this small chart here, this blue and curve show the bold response in one voxel inside the brain, and while from here to here is about 10 seconds. So this is based upon the fundamental physics of the bold response, and it cannot be simply changed by improving the speed of the functional MRI machine itself. There's another group of the activity called the electrophysiologic technology, and well known is the so-called EEG and MEG, or electroencephalography or magnetoencephalography, which measures electrophysiologic activity and neuronal activity. And it's really interesting that the EEG is very fast, as you can see here, this purple line, the very fast in time, by EEG and MEG is relatively low, is low in spatial resolution. So there's a lot of work have been done in the past decade to try to improve the EEG, MEG spatial resolution by solving something called electrical source imaging, or ESI, and furthermore pushing the limit of the resolution of spatial, temporal, and domain by integrating EEG, MEG with functional MRI. So I will touch on these two pieces of the work that we have been doing. One is try to map the brain dynamics from EEG recording, and then furthermore adding the functional MRI, try to improve the spatial temporal resolution.